Good morning, ladies and gentlemen. We are the 2017 NCTU Formosa Adjunct team. I'm Tiffany, and this is Justine. Allow us to introduce our project to you. This year, we focused on solving a serious agricultural problems in Taiwan, and we named our project AI Green Fingers. Hearing the name, you might think of our team project last year, peptide, which casts out from the past with spider venom peptides. So, what's new about our project this year? What is the difference? And what is the agricultural problem we're actually dealing with? Well, these questions can be answered by this chart. And the answer to the question is fungal diseases. And the bar graph shows the occurrence percentage of plant diseases in Taiwan. Apparently, we can observe that fungal diseases, the green part, account for almost 2/3 of them. Therefore, we can tell that the agricultural crisis brought by fungal diseases cannot be ignored, nor to be overlooked. Farmers always hope for the best farming situations that they hope their crops are free of fungal infection, but fungi exist everywhere. And if farmers surrender to the fungal diseases, myriads of damages and economical losses will be inevitable. So, what do farmers do to keep the fungal threat at bay? Nowadays, chemical pesticides are all they've got. They're cheap, they're effective, and they're easily accessible. Nevertheless, chemical pesticides are extremely harmful to the environment. Bioaccumulations, water pollution, soil pollutions, and the declining biodiversity are the drawbacks of using chemical pesticides. For the sake of protecting the environment, we should strive to avoid using them. But here's the thing. If we abandon using them, then how could we get rid of the fungal diseases? Well, we think biological pesticides will be the new alternative. Biological pesticides is a form of pesticides generated by microorganisms and other natural materials. And what's the exciting about that is that it avoids all the drawbacks chemical pesticides bring. But here comes another problem. When it comes to biological pesticides, effectiveness is doubtful for most of the people that it might not be more effective than the chemical ones, right? So, here comes our 2017 NCTU Formosa's team project. We filtered the most suitable biological pesticides with databases, just like using the big data analysis, which will be mentioned later. By using them, we will be able to select the most suitable biological pesticides from an innovative perspective. Coupled with synthetic biology, we may introduce a brand new biological pesticides that is eco-friendly, sustainable, and effective.
As for the databases we are using, there are several databases that might be available online. So, we introduced those databases into our project. The first databases we introduce is phytopath. Phytopath marks the relationship between plants and potential plant diseases. The second is phybase. Phybase enables us to know which pathogen will cause plant diseases. The third and the fourth databases are CNMP and PhytAMP. Oh no. Sorry. The third and the fourth databases are CAMP and PhytAMP. They illustrate the peptides that has been proved to have antifungal activity to a given pathogen. So through combining these four databases, we can know that the pathogen that will cause plant diseases, and the peptides that might be able to possess antifungal activity to a given plant, to a given pathogen. In order to find the most suitable antifungal peptides, we utilize the information we got from the aforementioned antifungal peptide databases. We try to categorize the peptides into several groups with the logic of its antifungal mechanisms. In this way, we can do experiments to evaluate the effectiveness of them. However, many peptides in the databases are just experimentally proven to have antifungal activity. The mechanism remains unknown. To make our process more feasible, we try to categorize those peptides by analyzing their sequences. The function of a peptide is often related to its sequence domain. Our strategy is to group the peptides by using CD search to find whether the peptides have conserved domains or not. Accordingly, we then get several groups that implies different conserved domains. To choose the best peptide candidates from each group, we try to categorize. We consider their sequence length, whether they're toxic to E. coli, whether E. coli can produce the peptides with correct folding structures, and their MIC values. Finally, we can filter out four peptides that in different groups from the databases with about 1,700 potential antifungal peptides. These following are these four peptides. First, MIMP1 from a MIMP family. The mechanism of the peptide is to bind specifically to beta-1,3 glucans on the cell wall of the cell membrane of the pathogen through conserved polar and aromatic residues. Second, LALTP from LTP family. LTP stands for lipid transfer protein. The mechanism of LTP is to bind to the lipid of the cell membrane of the pathogen and causes destabilization and permeabilization of the membrane. Third, RSAFP2, the mechanism from the Benson family. The mechanism of RSAFP2 is to interact with glycer, a kind of sphingolipids on the cell membrane of the pathogen. The interaction then induces a signaling cascade which ultimately causes cell death. Fourth, WITWIN1. WITWIN1 contains a borrowing domain which is related to the production of ROS in fungal hyphae. These four peptides will become our experiment targets.
And now all of you have an outline of how our project operates and how we choose our peptides. So allow me to show you the experiment we designed. For the experiment, we divided it into two parts. One, it's about producing our peptides via genetic engineering. And the other is examining the activity of the peptides through observing the result of hyphae, spore, as well as botany experiment. So for the first part, which is the peptides producing, we have chosen 4 targeting peptides, that is the MIMP1, WITWIN1, LALTP, and RSAFP2. And then we also chose 3 hosts, which correspond to each disease, and the hosts are orchid, tomato, and rice. Next, we assembled 4 targeting genes with the T7 promoters so that they can continuously produce the peptides, just like the ball breaks shown above. So far, you may have the doubts concerning our peptides' functionalities. So we do another experiment to examine the effectiveness of every peptide. So for the second part of our experiment, we will measure the size of the zone of inhibition, and we can compare the effect of the protein by the zone of inhibition. So for the experiment of the zone of inhibition, we will set the peptides on the white cotton sheets individually. That means the gray part, because the color problems. Okay, then we will set the pathogens in the middle of the plate. And then we will first measure the distance between the white cotton and the center of the pathogen, naming it A. After incubating the plate, we will then measure the distance between the edge of the inhibited hyphae and the center of the pathogen, and we will name it B. So the size of the zone can be calculated as the difference of A and B over A times 100%. We will do another experiment of IC50 value. IC50 is the half concentrations of the maximum inhibitory concentration. It represents the concentrations of the inhibitor which is required for 50% inhibition of fungus. So here's the step. Step 1, we will set the spores in the suitable temperature to germinate them. Step 2, we will add the spore suspension into the peptides in different concentrations. So the suspension without peptide will be the negative control. And step 3, we will repeat the step by adding 2 drops of 2 solutions on a microslide individually in different concentrations. Step 4, we will